Continuation of the previous video about the ribosomes. So structure, the ribosomes are spherical structures, not spherical but spheroid structures of 150 to 250 AO in diameter. Each ribosome is porous, hydrated and composed of two subunits. One ribosomal subunit is large in size and has a dome-like shape, while the other ribosome subunit is small in size and occurring above the larger subunit and forming a cap-like structure. This 70S ribosome consists of two subunits, 50S and 30S. The 50S ribosomal subunit is larger in size and has the size of 160 AO to 180 AO. The 30S ribosomal subunit is smaller in size and occurs above the 550 subunit like a cap. This 80S ribosome also consists of two subunits, 60S and 40S. The 60S ribosomal subunit is dome-shaped and larger in size, and the 40S ribosomal subunit is smaller in size and occurs above the 60S subunit, forming a cap-like structure. Both the subunits remain separated by a narrow cleft. The two ribosomal subunits remain united with each other due to high concentration of Mg2 plus ions. When the concentration of Mg2 plus ion reduces in the matrix, both ribosomal subunits get separated. During the protein synthesis, many ribosomes are aggregated due to common messenger RNA and form polysomes. A ribosome engaged in protein synthesis have four binding sites for the binding of mRNA and tRNA. They are M site for the binding of mRNA, A site, amino acyl site for the binding of incoming amino acyl tRNA, P site, peptidyl site for the binding of peptidyl tRNA, and E site for the binding of uncharged or unloaded tRNA. Large subunit, small subunit, large ribosomal subunit, E site, P site, mRNA binding site, small ribosomal subunit, etc. can be seen. Recently, following two models have been suggested to explain the three-dimensional structure of prokaryotic or 70S ribosome. Quasi-symmetrical model, according to the Stoffler-Rittmann quasi-symmetrical model, the larger subunit is almost bilaterally symmetrical and the smaller one irregular. The larger subunit has a spherical base and three protuberances. It is comparable to an armchair that has a circular seat. Its spherical base forms a seat for the smaller subunit. The prominent central protuberance forms the supportive back, and the lateral ones form the armrest. The smaller subunit is a slightly bent and bipartite rod comparable to a telephone receiver. It has a cleft which divides it into small head and large body or base. Then Lake's asymmetrical model. According to Lake's symmetrical model, both subunits are asymmetrical. The larger one is a spherical body or base and three major protuberances, and one central and others lateral. One of the lateral protuberances is long and slug-like, and the other one is only a ridge. The smaller subunit has an indentation which divides to an upper head and a lower body or base. Extending from the base is a small platform. Here you could see this picture. Head, base, cleft region, platform, central protuberance, head, platform, small subunit, large subunit, ribosome. Cleft, platform, head, base, small subunit, 30S subunit, ridge, valley, central protuberance, stalk, 50S subunit. Three-dimensional model of 80S ribosomes. Like 30S subunit of prokaryotes, the 40S subunit of eukaryotes head and base segments by transverse grooves. The 60S subunit is rounded in shape and its one side is flattened, and it becomes confluent with small subunit during the formation of monomer. Ribosomes have three binding sites for RNA polymerase molecules, one for mRNA and two for tRNA. One of the last two is P site and other is A site. Chemical composition. The ribosome is chemically composed of RNA and proteins as their major constituents. No lipid molecules. RNA and proteins. The 70S and 80S ribosomes, RNA and proteins present approximately in equal proportion, while 70S ribosomes contain more RNA than proteins, 60% to 40% than proteins. while 80S ribosomes contain less RNA than proteins. The 70S bacterial ribosome formed of nearly 54 kinds of proteins and 3 kinds of rRNAs. 30S subunit consists of 21 kind of protein and 16S rRNA, 5041 nucleotides, and 50S subunit consists of 34 kinds of protein, 23S rRNA. In 80S, 40S subunit formed of 30 to 40 kinds of protein and 18S rRNA. 18S rRNA, 21 proteins, 30S. Then 31 proteins, 50S, 70S ribosome. 18S rRNA, 13 proteins, 30 proteins, here 50 proteins. Functions. Ribosomes form the most important biosynthetic machinery for cells actively involved in protein production, provides the enzymes for protein synthesis, and serves as an ideal attachment site for the growing polypeptide chain. Ribosomal RNA probably act as a scaffold for holding proteins exactly in place. Ribosomes play a vital role in the catalytic role of ribosomes, and the ribosomal proteins enhance the activity of rRNA. Then constitution experiments of ribosomes. Reconstitution. To understand the three-dimensional organization of ribosomal proteins and also for the investigation of interaction between molecules of rRNA and proteins, certain kinds of approaches have been adapted in recent times. The approach was put forward by Nomura. He purified 30S ribosomal subunit and dissociated them chemical means into their components, RNAs and proteins. They allow them to reassociate under appropriate ionic conditions. The reconstitution of ribosomes have been a dramatic advance of recent times. When ribosomes are centrifuged in a gradient of 5 mCSCl, about 30 to 40% of the proteins is lost. Both the subunits dissociate into two inactive core particles which contain the RNA and some proteins, core proteins. Simultaneously, some other proteins called split proteins are released from the ribosomes. When the split proteins are added to their corresponding core particles, the ribosome is reconstituted. The reconstitution of functional 30S subunit has been achieved by the addition of 16S rRNA to a mixture of proteins from the 30S subunit. Following a similar procedure, the 50S subunit has also been reconstituted along with the subunits of the whole ribosome. It is also assembled. When the 30S subunit is treated with 5 urea and 2 molar lithium chloride, it dissociates and releases all the proteins. The 60S rRNA can be extracted from the 30S subunit by phenol. If now the 16S RNA is put together with the proteins and the same subunit, reconstruction takes place. The reconstitution of 30S ribosomes take place in 2 steps. In the first step, carried out at low temperature, 60S RNA binds with 30S ribosomal proteins and forms R1 particles. Reconstitution intermediate. In the second step, the R1 intermediate are heated at 40 degrees Celsius in the presence of other proteins that are remained in the supernatant and forming an excited intermediate. With the 20 minutes fully active 30S is formed, the self-assembly of 30S ribosomes is highly specific. The reconstitution of ribosomal components into complete ribosomes has been observed in E. coli. Several small precursor forms have been found in E. coli cells before final mature subunits are formed. Mutant strains of E. coli have been selected by U3 and M. Nomura by blocking at several discrete stages of self-assembly. Therefore, accumulate smaller precursor forms. Such mutations would be lethal since only mature ribosomal subunits can participate in protein synthesis. The optimal rate of self-assembly occur in the cell at 37 degrees Celsius. If any ribosomal proteins and rRNAs carry mutations, could be deliberating in the code. The study of defects carried by SAD strains also help in understanding interactions between rRNAs and proteins. Nomura's great experiments made with antibiotics given like the molecular structure of ribosomes. Streptomycin resistant strains of E. coli was found to carry an alternation in single ribosomal protein 5p10. To prove that the altered p10 protein is responsible for the altered response to streptomycin P10D proteins isolated from a streptomycin sensitive strain and used to reconstitute 30S subunit in which all other proteins and RNA are derived from resistant strains. The reciprocal experiment was also performed by using P10 proteins from streptomycin resistant strain and other components from a sensitive strain. The result was clear. Reconstructed subunit with the sensitive P10 proteins were streptomycin sensitive, even though all other proteins and RNA come from resistant ribosomes. The resistant P10 proteins conferred resistance in vitro, since the binding of streptomycin to ribosome is interference with current reading frame of genetic code. The P10 proteins is present at or near the ribosomal site at which reading process occurs. In such manner, the functions of numerous proteins and RNAs of ribosomes will be elucidated. Molecular mechanisms involved in recombination of DNA. Next topic. So, recombination. Two DNA molecules exchange genetic information, resulting in the production of new combination of alleles. This recombination process creates genetic diversity at the level of genes that reflect differences in the DNA sequences of different organisms. In eukaryotic cells, which are cells with the nucleus and organelles, recombination typically occurs during meiosis. During the alignment, the arms of the chromosomes can overlap and temporarily fuse, causing a crossover. Crossovers result in recombination and the exchange of genetic material between the maternal and paternal chromosomes. As a result, offspring can have different combinations of genes than their parents. Then genetic recombination. Evolution could not happen without genetic recombination. If it were not possible to exchange materials between chromosomes, the content of each individual chromosomes would be irretrievably fixed in its particular alleles. When mutations occur, it would not be possible to separate favorable and unfavorable changes. The length of the target for mutation damage would effectively be increased from the gene to the chromosome. Ultimately, a chromosome would accumulate so many deleterious mutations that it would fail to function. By shuffling the genes, recombination allows favorable and unfavorable mutations to be separated and tested as individual units in new assortments. It provides a means of escape and spreading for favorable alleles and a means to eliminate an unfavorable allele without bringing down all the other genes with which this allele is associated. This is a basis for natural selection. Recombination occurs between precisely corresponding sequences so that not a single base pair is added to or lost from the recombinant chromosome. Homologous recombination. Recombination involves reaction between homologous sequences of DNA. Is called generalized or homologous recombination. It is a physical phenomenon where exchange of sequence occurs with no net gain or loss of nucleotides. This is based on sequence complementarity. Occurs between sequences that are nearly identical. It is the most important version of recombination in nature, being responsible for meiotic crossing over in eukaryotes and the interaction of acquired DNA by the process of conjugation, transduction, transformation into bacterial genomes. It involves a reciprocal exchange of sequence of DNA. In eukaryotes, it occurs at meiosis, usually both in males during spermatogenesis and females during oogenesis. Parental or paternal chromosome, maternal chromosome, DNA replication, crossing over. So, paternal, maternal crossing over resulting recombined chromosome. Homologous recombination is a reaction between two duplexes of DNA. Its critical feature is that the enzymes responsible can use any pair of homologous sequences as substantial, although some types of sequences may be favored over the other. The frequency of recombination is not constant throughout the genome, but is influenced by both global and local effects. Homologous recombination is extensively studied in E. coli. At least 25 proteins are involved in recombination in E. coli. Recombination occurs during protracted prophase of meiosis. Progress through meiosis. Leptothene condensed chromosome become visible, often attached to nuclear envelope. Nuclear interaction. Each chromosome has replicated and consists of two sister chromatids. Initiation, strand exchange, single strands exchange, assimilation, region of exchange strands is extended, then resolution. The beginning of the meiosis is marked by the point at which individual chromosomes are visible. Each of these chromosomes have replicated previously and consist of two sister chromatids, each of which consists of a duplex DNA. The homologous chromosomes approach one another and begin to pair in one or more regions, forming bivalence. Pairing extends until the entire length of each chromosome is opposed with its homologue. This process is called synapsis or chromosome pairing. When the process is completed, the chromosomes are laterally associated in the form of synaptoanymal complex. Recombination between chromosomes involves a physical exchange of parts, usually represented as a breakage and reunion, in which two non-sister chromatids have been broken and then inked with each other. When the chromosomes begin to separate, they can be seen to be held together at discrete sites called chiasmata. Recombination requires a mechanism that allows the duplex DNA of one sister chromatid to interact with the duplex DNA of a sister chromatid from the other chromosomes. It must be possible for this reaction to occur between any pair of corresponding sequence in the two molecules in a highly specific manner that allows materials to be exchanged with precision at the level of individual base pair. We know of only one mechanism for nucleic acid to recognize one another on the basis of sequence complementarity between single strands. The first step in providing single strands is to make a break on each DNA duplex. One or both of the strands of that duplex can then be released. If at least one strand displaces the corresponding strand on the other duplex, the DNA duplex molecule will be specifically connected and corresponding sequence. If the strand exchange is extended, there can be more extensive connection between the duplexes. By exchanging both strands and later cutting them, it is possible to connect to the paternal duplex molecule by means of crossover that corresponds to the demands of a breakage and reunion. The key event in recombination between two duplex DNA molecule is exchange of a single strands. When a single strand from one duplex displaces the counterpart in the other duplex, it creates a branched structure. The exchange generates a structure of heteroduplex DNA consisting of one strand from each parent. Two reciprocal exchanges are necessary to generate joint molecule. The joint molecule is resolved into two separate duplex molecules by nicking two of the connecting strands. Whether recombinants are formed depends on whether the strands involved in the original exchange or the other type of strands are nicked during resolution. So recombination during meiosis is initiated in double strand breaks. A likely pathway for homologous recombination during meiosis is outlined in 3235E earlier. So the model has four key features. First, homologous chromosomes align. Second, a double strand break on DNA molecule is created and the exposed ends are processed by an exonuclease, leaving a single stranded extension with a free 3' hydroxyl group at the broken end. And third, the exposed 3' end invade the intact duplex DNA of the homologue and is allowed by branch migration or replication to create a pair of holiday-mediates. Fourth, cleavage of the two crossover creates either of two pairs of complete recombinant products. In this double strand break pair model for recombination, the 3' ends are used to initiate a genetic exchange. Once paired with the complementary strand on the intact homologue, a region of hybrid DNA is created that contains complementary strands from two different parent DNAs, the product of step 2. Each of the 3' ends can then act as a primer for DNA replication. Meiotic homologous recombination can vary in many details from one species to another, but most of the steps outlined above are generally present in some form. There are two ways to resolve the holiday intermediate with RUVC-like nucleases so that the two products carry genes in the same linear order as in the substrate, the original and unrecombined chromosomes. If cleaved one way, the DNA flanking, the origin of the region containing the hybrid DNA is not recombined. If cleaved the other way, the flanking DNA is recombined. Both outcomes are observed in vivo. Bacterial homologous recombination is a DNA repair function. In bacteria, homologous genetic recombination is a primarily DNA repairing process and is referred to as a recombinational DNA repair. It is usually directed at the reconstruction of replication forks that have started or collapsed at the site of DNA damage. Homologous genetic recombination can also occur during conjugation or mating. When chromosomal DNA is transferred from one bacterial cell to another, recombination during conjugation, although rare in wild bacterial population, contributes to genetic diversity. An example of what happens when a replication fork encounters DNA damage. A common feature of DNA repair pathways is that they introduce a transient break into one of the DNA strands. If a replication fork encounters a damaged site under repair near a break in one of the template strands, one arm of the replication fork becomes disconnected by a double strand break on the fork collapse. The end of the break is processed by degrading the 5' ending strand. The resulting 3' single-stranded extension is bound by a recombinase that uses it to promote strand inversion. The 3' end inverts the intact duplex DNA connected to the other arm of the fork and pairs with its complementary sequence. This creates a branched DNA structure, a point where 3 DNA segments come together. The DNA branch can be moved in a process called branch migration to create an X-like crossover structure known as Holliday intermediate, named after researcher Robin Holliday, who first postulated this existence. The Holliday intermediate is cleaved or resolved in a special class of nuclease. The overall process reconstructs the replication fork. The E. coli. In E. coli, the DNA end processing is promoted by REC bed nuclease or helicase. The REC-PCD enzyme binds to linear DNA atom free broken end and moves inward along the double helix, unwinding and degrading the DNA in reaction coupled to ATP hydrolysis. The REC-B and REC-D subunits are helicase motors, with REC-B moving 3' to 5' along one strand and REC-D moving 5' to 3' along the other strand. The activity of the enzyme is altered when it interacts with sequence referred to as Chi, which binds tightly to a site on the REC-C subunit. From that point, degradation of the strand with the 3' terminus is greatly reduced, but the degradation of the 5' terminal strand is increased. This process creates a single-stranded DNA with a 3' end, which is used during subsequent steps in recombination. Chi-square scattered throughout the E. coli genome enhances the frequency of recombination about 5 to 10-fold within 100 bp of Chi site. The enhancement declines the distance from Chi increase. Sequences that enhance recombination frequency have also been identified in several other organisms. The bacterial recombinase is the REC protein. REC-A is unusual among the proteins of DNA metabolism in that its active form is an ordered helical filament and up to several thousand subunits that assemble cooperatively on the DNA. This filament usually forms a single-stranded DNA. Site-specific recombination. A region of extension homology is not a prerequisite for recombination. The process can also be initiated between two DNA molecules that have only very short sequences in common. This is called site-specific recombination. This was first characterized in prokaryotes, where specialized recombination, also known as the site-specific recombination, is responsible for integration of phage genomes into the bacterial chromosomes. The recombination event involves specific sequences of phage DNA and bacterial DNA, which include a short stretch of homology. The enzymes involved in this event act only on the particular pair of target sequences in an intermolecular reaction. The enzymes that catalyze site-specific recombinations are generally called recombinases. Those involved in phage integration are related to those enzymes and also known as the integrase family. Prominent members of the integrase family are the prototype INT. Integration and excision occur by recombination of specific loci on the bacterial and phage DNA called attachment sites. The attachment site on the bacterial chromosome is called at lambda in bacterial genetics. Lambda ATT. for describing the integration or excision reaction. The bacterial attachment site is called at B, consisting of the sequence components BOB. The attachment site on the phage ATTP consists of an component POP. The sequence 0 is common to ATTP and ATTP. It is called ATTB and ATTP. It is called core sequence and recombination events occur within it. The phage DNA is circular, so the recombination event inserts it into the bacterial chromosome as a linear sequence. The prophage is bounded by two new ATT sites, the production of the recombination called ATTL and ATTR. An important consequence of the constitution of ATT site is that the integration and excision reactions do not involve the same pair of reacting sequences. Integration requires recognition between ATTB and ATTP, whereas excision requires recognition between ATTL and ATTR. The difference in the pairs of sites reacting in integration and excision is reflected by a difference in the proteins that mediate the two reactions. Integration requires the product of phage gene INT, which codes for an integrase enzyme and bacterial protein called integration host factor. Excision requires the product of phage gene XIS in addition to INT and IHF. Some double-strand breaks are repaired by renal homologous end joining. Double-stranded breaks sometimes occur when recombinational DNA repair is not feasible, such as during phase of cell cycle when replication is occurring and known cystochromatids are present. At these times, another path is needed to avoid the cell death that would result for a broken chromosome. That alternative is provided by homologous end joining. Next topic, genetic code. Genetic code is a nucleotide sequence of DNA and mRNA that specifies the amino acid sequence of proteins. During protein synthesis, genetic code is first copied down from DNA to mRNA. This is called genetic transcription. In the next stage, it is transferred from mRNA to protein. It is called genetic translation. Genetic code is visualized as a complex coding system composed of a series of coding units called codons. A codon is a nucleotide triplet formed of a set of 3 nucleotides. It is called a triplet codon. It codes 4 specific amino acids. The sum total of the codons required for the synthesis of a polypeptide chain or the protein molecule is called a cistron or gene. The sum total of the codons required for the synthesis of a polypeptide chain or a protein molecule is known as cistron or gene. The DNA molecule can contain many such cistrons. The 4 kinds of nucleotides of DNA or mRNA can enter into triplet combinations and form as many as 64 kinds of codons. This code only for 20 kinds of proteogenic amino acids. So it becomes clear that some codons never code for amino acids, and at the same time some amino acids are coded by more than one codon. Those codons which code for amino acids are known as sense codons. Those which do not code for amino acids were formerly called nonsense codons. Characteristics of genetic code. The code is a triplet codon. The nucleotides of mRNA are arranged as a linear sequence of codons, each codon consisting of 3 successive nitrogenous bases. Code is a triplet codon. If codons are presumed to be stringlets, the 4 types of nucleotides can form only 4 codons, which in turn can code for only 4 amino acids. If codons are visualized as doublets, the 4 types of nucleotides can form as many as 16 codons, which can code for only 16 amino acids. However, if the codons are considered to be triplets, there would be as many as 64 codons, which are more than enough to code for 20 proteogenic amino acids. Thus, it becomes all the more explicit that genetic code consists of triplet codons and each codon is a trinucleotide sequence. The codon is non-overlapping. The nucleotide of a codon do not normally overlap the nucleotides of an adjacent codon. In translating mRNA molecules, the codon do not overlap but are read sequentially. The code is commaless. The genetic code is commaless, which means that no codon is reserved for punctuations. It means that after one amino acid is coded, the second amino acid will be automatically coded by the next 3 letters, and that no letters are wasted as the punctuation mark. The code is non-ambiguous. Non-ambiguous code means that a particular codon will always code for the same amino acid. In case of ambiguous code, the same codon could have different meanings, or in other words, the same codon could code for 2 or more than 2 different amino acids. The code has polarity. The code is always read in a fixed direction, that is in the 5' to 3' direction. In other words, the codon has a polarity. It is apparent that if the code is read in opposite direction, it would specify 2 different proteins. The code is degenerate. More than one codon may specify the same amino acid, that is called degeneracy of code. For example, except for tryptophan and methionine, there are single codons, each have a single codon each. All other 18 amino acids have more than one codon. These 9 amino acids, namely phenylalanine, tyrosine, histidine, glutamine, etc., have 2 codons. Isoleucine has 3 codons. Genetic code is considered degenerate because one amino acid may be specified by more than one codon. The collaboration of more than one codon to specify the amino acids is called code degeneracy. Some codes act as start codons. In most organisms, AUG codon is the start or initiation codon, that is, the polypeptide chain starts either with methionine eukaryotes or N-formyl methionine prokaryotes. Methionine and N-formyl methionine tRNA specifically binds to the initiation site of mRNA containing the AUG initiation codon. In rare cases, GUG also serves as an initiation codon. Bacterial protein synthesis. Normally, GUG codes for valine, but when normal AUG codon is lost by deletion, only the GUG is used as initiation codon. 3 codons. Some codes act as stop codons. 3 codons UAA, UAG and UGA are the chain stop or termination codons. They do not code for any of the amino acids. The codons are not read by any tRNA molecules but are read by some specific proteins called release factors. RF1, RF2, RF3 in prokaryotes and RF in eukaryotes. These codons are also called nonsense codons since they do not specify any amino acids. The code is universal. Genetic code is considered universal. This means that the same code is present in all the organisms and also in the code specifies the same amino acid in all organisms, right from the simplest virus down to the genetic code was first established by Marshall and Nirenberg. Start codon and stop codon. Start codon. Start codon is the first codon of the transcribed mRNA sequence that translates into an amino acid by ribosome. Therefore, it marks the site at which translation into a protein begins. It is a sequence composed of 3 nucleotides. During translation, tRNA recognizes the start codon and begins the translation. AUG is the most common start codon. In eukaryotic genes, it specifies the amino acid methionine, while the prokaryotic gene, it specifies the formyl methionine, F-met. However, there are some alternative start codons in both eukaryotes and prokaryotes, which usually code for amino acids other than methionine, but non-AUG start codons are rarely found in eukaryotes. Some alternative start codons are CUG, AUE and AUU in humans and GUG and UUG in prokaryotes. Then stop codon. The stop codon is a trinucleotide sequence which marks the site at which translation of the mRNA into protein ends. Hence, it is the last codon of transcribed mRNA. There are 3 stop codons: amber, opal or umber and ocreal. No, not that. UAG, UGA and UAA. They signal the halt to protein synthesis. They are also known as termination codons or nonsense codons. They do not code for an amino acid. During the transition, stop codon is responsible for releasing the newly formed polypeptide chain from the ribosome. Since no tRNA exists with the anticodon complementary to the stop codon. Similarities between start codon and stop codon. Start and stop codons have 2 punctuation marks in the genetic code. They are the 2 punctuation marks in the genetic code. They are trinucleotide sequences within a messenger RNA. Both codons are important to minimize coding error. Then differences between start codon and stop codon. Start codon is the codon that marks the site at which translation begins, while the stop codon is the site at which translation stops. So it is the key difference between these two. Start codon is found at the 5' end of the mRNA, while stop codon is present at the 3' end of the mRNA. Moreover, start codon codes for methionine, while stop codons do not code for amino acids. Degeneracy of the code, Wobble hypothesis and isoacceptor tRNA. Wobble hypothesis. There are more than one codon for one amino acid. This is called degeneracy of genetic code. To explain the possible cause of degeneracy of codons, in 1966, Francis Crick proposed the Wobble hypothesis. According to this hypothesis, only the first 2 bases of the codon have a precise pairing with the bases of anticodon of tRNA, while the pairing between the third bases of codon and anticodon may wobble. Wobble means to sway or move unsteadily. The phenomenon permits a single tRNA to recognize more than one codon. Therefore, although there are 61 codons for amino acids, the number of tRNA is far less, around 40, which is due to wobbling. The wobble hypothesis states that the base at 5' end of the anticodon is not specifically confined as the other two bases, allowing it to form hydrogen bonds with any of several bases located at the 3' end of a codon. This leads to the following conclusion. The first two bases of the codon makes normal H bond pairs with second and third bases of the anticodon. At the remaining position, less stringent rules apply and non-canonical pairing may occur. The wobble hypothesis thus proposes a more flexible set of base pairing rules at the third position of the codon. The relaxed base pairing requirement or wobble allows the anticodon of a single form of tRNA to pair with more than one triplet in mRNA. The rule first base U can recognize U or C and first base I can recognize U, C or I. A. Crick's hypothesis hence predicts that the initial two ribonucleotides of triplet codes are often more critical than the third member in attaching the correct tRNA. Wobble base pair. The wobble base pair is paired in between two nucleotides in RNA molecules that does not follow Watson-Crick base pair rules. The four main wobble base pairs are guanine uracil, hypoxanthine uracil, hypoxanthine adenine, hypoxanthine cytosine. In order to maintain consistency of nucleic acid nomenclature, I is used for hypoxanthine because hypoxanthine is the nuclear base of inosine. Inosine displays the true qualities of wobble in that if that is the first nucleotide in the anticodon, then any of three bases in the original codon can be matched with the tRNA. Isoacceptor tRNA or transfer RNA is a type of RNA which help in the synthesis of protein from mRNA. tRNA functions as adapter molecule during the translation process. It was earlier known as soluble RNA or siRNA. As an adapter, it links the amino acids to nucleic acid. If it carrier the amino acid to be added in the peptide chain and also deciphers the codon for the same in mRNA molecule. Multiple tRNA representing the same codon are called isoaccepting tRNA or cognate tRNA. Acceptor stem and anticodon stem make tight contact with synthetase and mutase in this region, disrupt this contact. Isoaccepting tRNAs are charged by same single amino acid tRNA synthetase. All tRNAs are divided into groups and each group identifies its particular synthetase. tRNAs are recognized by synthetase by small number of bases. Next topic, transposons in eukaryotes. Topic sign, alien family, line N1. P elements in Drosophila, transposons in maize. Transposons. Transposable elements can be defined as small mobile DNA sequences that move around chromosomes with no regard for homology. Transposable elements are DNA segments which can mobilize them from their original location and insert into new positions in the genome. These occur in almost all genomes. Half of the human genome consists of transposable elements, and large plant genomes are composed of more than 80% of transposable elements. Prevailing among these are the retrotransposons that propagate through an RNA intermediate. DNA transposons move on DNA by cut and paste mechanism. The vast majority of transposons insertion into genes result in gene damage. This is therefore mutual interest of host cells and transposons to limit transposition to very low frequencies. Environmental stress like genome shock can lead to release of epigenetic silencing and reactivation of transposons. So transposon insertion into gene are almost deleterious, but in rare cases can result in novel gene regulation patterns. So very rarely transposons were domesticated to provide novel functions to the host. Transcriptions can be applied in genetic tools for mutagenesis and gene isolation. These elements are discovered by Barbara McClintock in maize. She discovered mobile DNA in maize, and the mobility of these elements caused different maize kernels used by Heads and Jacob. So transposons are the segment of DNA that move from one location to another in a chromosome or another chromosome, which is excised one site and inserted to another site, jumping around the genome that causes mutations and change the amount of DNA in the genome. They have unique capability to duplicate themselves and not duplicated by transferring themselves. Transposons are grouped together on the type of element, structure, and mechanism of transposition. Viral transposons, retrovirus, Drosophila yeast, and non-viral transposons, eukaryotic non-LTR. Then bacterial transposons, Drosophila, maize, etc. Then sign, short interspaced nuclear elements. Signs are groups of non-LTR and non-autonomous retrotransposons. They are small bases found in very diverse eukaryotes. They typically possess a head with RNA polymerase 3. Sign do not decode a functional reverse transcriptase protein. Sign DNA RNA forms complex with line ORF2 proteins and inserted genomes by creating short RSV insertion. The internal region of signs originate from tRNA and remain highly conserved, suggesting positive pressure to preserve structure and function of signs. While signs present in many copies of vertebrates and invertebrates, signs are often lineage specific, making them useful markers of divergent evolution between species. Mutation in sign sequence make it possible to construct phylogenes based on differences in sign between species. They are also implicated in certain types of genetic diseases in human and other eukaryotes. There are 50 human diseases that have associated in sign. Two types, ALU and B1. ALU family, short length of DNA, originally characterized by the action of Anthropobacter eluteluteus restriction endonuclease, which are most abundant in transposable element containing over 1 million copies dispersed throughout the human genome, so known as selfish or parasitic genes. Study of ALU elements has also been important in elucidation of human population genetics and the evolution of primates, including the evolution of humans. The most common sign in primates is ALU. They are approximately 350 base pair long and not contain any coding sequence and can be recognized by the restriction endonuclease line. Long interspaced nuclear element. Group of genetic elements that are found in large numbers in eukaryotic genome, that are group of non-LTR retrotransposons. They make up around 21.1% of human genome and make up a family of transposons. Each line is about 7,000 base pair long. Lines are transcribed into mRNA and translated into protein that act as reverse transcriptase. They make a DNA copy of line RNA that can be integrated into a genome at a new site. The only abundant DNA in human is line 1. One genome contains 100,000 translated and 4,000 full-length L1 elements. Comparison with line DNA sequences can be used to the transposon insertion in the genome. P elements in Drosophila. Egg, sperm, egg, sperm. P elements are repressed. P elements are activated. P elements are transposable elements that were discovered in Drosophila in the causative agents of genetic traits called hybrid dysgenesis, which means annotation or aberrant mutation, chromosome breakers and mutation. Transposons are responsible for P trait of P element, and it is found only in wild flies. All P elements have a canonical structure containing 31 base pair terminal inverted repeats and 11 base pair terminal inverted repeats located at TH4P domain of transposase. The shortest and longest P element are non-elements. In hybrid dysgenesis, one strain of Drosophila mates with another strain of Drosophila and producing hybrid offspring cause chromosomal damage known to be dysgenic. Hybrid dysgenesis requires a contribution for both parents. For example, in PM system, P strain contributing paternal and M strain contributing maternal. The reverse cross, the M father and P mother produces normal offspring. P male chromosome can cause dysgenesis when crossed with M female. P element also encodes the suppressor of transposition, which accumulates in the cytoplasm during the development of cells. Thus, in a crosses of P or M male at the period male, the female cytoplasm contains suppressor which binds to elements and prevents their transposition. P elements are commonly used as mutagenic agents in genetic experiments in Drosophila. An advantage of this approach is that the mutation can easy to locate. Dysgenic cross and reciprocal cross. Transposons in maize. Early experiment of Rollins Emerson and the maize genes that cause multicolor or variant kernels. Most core kernels are either wholly pigmented or colorless, but Emerson noted that some yellow kernels had spots or different color. He proposed these kernels are resulted to form an unstable mutation on chromosome. Yet Emerson had not provided for the explanation of the phenomenon. M.C. Clinton instead suggested that the occurrence of an unexpected purple and brown colored kernels was due to genetic transposition. C gene, wild type, pigmented, no AC, colorless, spotted kernels. To test this claim, he bred maize plant to vary in the color of the kernels. M.C. Clinton took cell samples from the corn and dyed the samples, looked at them under the microscope by using staining techniques to enhance the contrast in the microscopic images and observe chromosomes. He bred maize plants carrying chromosomes that resulted in the recessive brown phenotype called BZ. Why are these kernels spotted? The amount of brown spots on the creamy background depend on the timing of breakage during development. M.C. Clintock's experiment on maize plant highlighted the instability of genetic material and started the existence of transposable elements in the genome. 4D element transposing under influence by 4C element insert into allele that destroying the ability to produce pigment. After the transposition of DS into the kernel, this kernel will be colorless. Normal C gene expressing pigmented product. Disrupted gene, normal C gene.